Hi everyone. Today I would like to share with you an abstract presentation entitled on molecular characterization of the cultural aerobic bacteria in the midgut of the field, caught in Culex and different Culex species in Sri Lanka. So therefore, let's have a look, the importance of this study. That is, the first part gonna be the background. So in this case, the larva and the adult mosquito stages, they can harbor different extracellular microbes, which are exhibiting various function in their digestive tracts, including the host parasite interaction. So normally this, the midgut symbiotic bacteria, which is genetically exploited to express molecules within the vector. And this is really very important for altering the vector competency. That means, the ability of the vector to transmit a pathogen. And also it will be very important, for disease transmission. It will have a potential for disease transmission. Therefore, if we are trying to if we are trying to identify the mosquito gut inhabiting the microbiota, it will give us an apple of importance before developing a novel vector control strategy, which enables for the modification of the vector. So therefore, how was the method of the study? Well, they collect the adult mosquito. So they did collection of the adult mosquito, different colexes, and then in Sri Lanka, and then after the midgut was lysate of the field caught on non-blood fetus female mosquito were cultured, then they cultured, and then the prokaryotic 16S ribosomal RNA partial genes of the isolated bacteria colony were amplified, followed by DNA sequencing. So later, with by targeting the 60S and ribosomal RNA partial genes, they did the isolation, and then finally they did the DNA sequencing. So therefore, the diversity in the index were used to us as a diversity and the richness of the bacteria isolate in the three mosquito species. So the distribution pattern of the bacterial isolate between the different mosquito species was assessed by the distance-based redundancy analysis. So what was the result of this? Well, the result was a total of 20 bacterial species, different bacterial species like the Staphylococcus, the Bacillus, the Staphylococcus, different species, the Bacilla, Bacillus, Staphylococcus, the Moraxella as well, the Enterobacter, the Klebsiella, almost different, the Streptomyces, so were identified, right? And all of these species belong to the three phylum. So the Proteobacteria, the Firmicutes, and the Actinobacteria, out of which the phylum Firmicutes, which is accounting 71%, was the most prominent in the study. So the least number of the species was recorded from Actinobacteria. So therefore, the relative distribution of the midgut microbiome in different mosquito species differs significantly among the mosquito species. Therefore, when we look the midgut microbiomes among the different mosquito species, it was significantly different. So the midgut microbiota of the Culex indicate a similarity of 21%, while for this they share 6 point. So with a cluster of above two species. Therefore, the gut microbiota of the Culex was also significantly more diverse and more evenly distributed compared to the other mosquito species. Therefore, the symptom diversity, the molecular diversity, were higher. So of this recorded species is, the Bacillia different can be recommended as a potential candidates for the paratransgenesis. So what will be the conclusion of such kind of study? So therefore, the relative distribution of the midgut microbiome in different mosquito species differs significantly among the three studies adult mosquito species. So the present data strongly encourage further investigation to explore the potential usage of these microbiomes through the paratransgenic approach for the novel eco-friendly vector control strategy. So this is the abstract presentation from Sri Lanka study. So therefore, in conclusion, this study indicated that when we have the mosquito species, different mosquito species, their microbiome is significantly different, right? So this is another most important part that I would like to tell for you for the today's lesson. And the dominant was on the three species that was from the protobacteria and the Firmicutes and the actinobacteria, but the Firmicutes was the most prominent from this study. So thank you for watching, and if you like it, just subscribe and share to your friends.